identify different kinds of research fields and interests and specialities that they may wish to pursue in the future. So today we will be going through heart failure, and this is an opportunity to sort of be introduced to the current advancements with different types of research that is currently taking place within the field, and maybe future directions that you may be interested in pursuing in the future. So today, Young Academics as well, we don't just run Researcher Path, we also help students find research opportunities, help them get published in research and books, and also help students find unique opportunities within business or arts, kind of marrying medicine with different types of careers outside of medicine as well. So if you're interested, please do look on our social media for other types of events and opportunities. Today, I'd like to introduce Dr. Oceandy, who is a scientist in cardiovascular sciences at the University of Manchester, and his laboratory focuses on the molecular aspects of heart failure and studying novel approaches to repair and regenerate damaged hearts. His work is supported by multiple different organisations, including the British Heart Foundation, the Medical Research Council, and Heart Research UK. Neve is a soon-to-be third-year medical student with a strong interest in cardiology and cardiothoracic surgery. Between studying, working as a bartender, and a committee role in the Women in Medicine Society, she can be found on Instagram promoting a healthy lifestyle and mental well-being at healthy.be. So over to you, Neve.
So hi everyone, I'm going to be doing the first part of the talk, so I'm just going to quickly share my slides. Can you see that, everyone? Okay, so I'm just going to be covering some ideas such as cardiac plasticity, its relevancy to hypertrophy and consequential heart failure, and kinase signalling as a targetable mechanism for the treatment of heart failure. So it's first important to understand that the heart is capable of remodelling, something referred to as cardiac plasticity. Throughout a person's life, they may be exposed to a variety of environmental conditions, and these can have an impact on the heart structure. For example, in periods of prolonged bed rest, or the use of a ventricular assist device, the heart can atrophy due to lower demands. In opposition to atrophy, the heart can also hypertrophy. In pregnancy, or with regular exercise, the heart will undergo physiologic hypertrophy to meet increased oxygen demands. In cases such as hypertension, the heart will undergo pathological hypertrophy as the heart has to pump against a higher pressure. Whilst physiological hypertrophy is advantageous, pathological hypertrophy can increase the risk of ventricular dilation, leading to heart failure and ventricular arrhythmia. You can see that these two cases of hypertrophy look quite different, as you can see in the diagram, and they have different effects. This is because there's different mechanisms involved in the development of these structural changes, particularly in kinase signalling. So hypertrophic growth is the primary mechanism in which the heart reduces stress on the ventricular wall. Predictably, this involves an increase in protein synthesis, as well as the size and organization of sarcomeres, which are the force-generating units in individual myocytes. The response of the heart to physiological demand can be really quite remarkable, with the left ventricular mass in athletes being around 60% greater than in non-athletic controls. And in many heart diseases, there's similar effects with pathological hypertrophy. So with all of this taken into account, along with atrophy potential, it can be seen that the heart has a dynamic growth range of around 100%, which is very, very large. So hypertrophy can occur in two ways, either concentric or eccentric. And in concentric hypertrophy, the sarcomeres are added in parallel to create a thicker heart wall that is able to cope with the chronic pressure overload in cases such as hypertension. This structure reduces the volume of the heart chamber as the radius is unchanged. In this scenario, there continues to be a pressure overload as hypertension continues, and now there's an additional volume overload due to the impaired filling ability of the chamber. So now sarcomeres begin to be added in series instead of in parallel, and this is known as eccentric hypertrophy. This increases the radius of the chamber and leads to ventricular dilation. We can see in this diagram a depiction of a heart with pathological hypertrophy. In this case, the myocyte width exceeds the myocyte length as sarcomeres have been added in parallel. As sarcomeres begin to be added in series, the myocyte length begins to exceed myocyte width, and as shown, causes cardiac dilation, leading to advanced cardiac dysfunction as the dilated ventricle has elevated wall stress and oxygen demand, but a lower mechanical efficiency. And clinically, this is associated with symptoms and progression of heart failure. So the two major triggers of cardiac hypertrophy are biochemical and neurohumoral stress. These induce intracellular signaling cascades that ultimately lead to an increase in protein synthesis, stability, or both, leading to an increase in cardiomyocyte size, i.e. hypertrophy. If you take a look at the diagram on the right, you can see that the signals are first transduced at the cell membrane. In the case of biochemical stress, these mechanisms are not well understood, but probably involve stretch-sensitive ion channels and integrins, along with other structural proteins that link the extracellular matrix, the cytoskeleton, the sarcomere, and calcium-handling proteins, along with the nucleus in a complex network. Signals from neurohumoral stress, as well as potent growth factors, are similarly transduced at the cell membrane, also leading to an intracellular cascade that ultimately communicates with the nucleus to bring about hypertrophic gene expression. Many of these signaling molecules involved are calcium dependent, making intracellular calcium homeostasis a proximal trigger in stress-induced remodeling. Hypertrophic stimuli are transduced by many calcium-dependent molecules, including mitogen-activated protein kinase, known as MAPK, which we'll talk about in the next few slides. Calcineurin is a particularly potent inducer of hypertrophy, and in many cases is sufficient or even necessary for pathologic hypertrophy. These are just a few of the mechanisms involved, and so it can be seen that there is a complex network of signaling pathways involved in pathological hypertrophy that give a limited number of responses that constitute hypertrophy, as well as myocardial damage with fibrosis and cell death. In contrast, one major pathway in physiological hypertrophy is the PI3K-AKT pathway, although long-term stimulation of this can also lead to pathological hypertrophy. So in this slide, we can see a bit of a scary looking diagram of some of the signaling pathways that are involved in cardiac hypertrophy. Clearly, the mechanism is very vast and complicated, but we can see some molecules that we've already discussed, such as the PI3K-AKT pathway that's involved in physiological hypertrophy, and calcineurin, which is highly involved in pathological remodeling. However, I want to now focus on these molecules that are underlined in red. These are mitogen-activated protein kinases, or MAPKs. As you can see, they're stimulated by stress, which then triggers an intracellular signaling cascade that ultimately leads in the activation of transcription factors, particularly that go on to alter gene expression, particularly upregulating cardiac hypertrophy. I also want to bring attention to this molecule, RAS, which is an upstream regulator of many of these signaling pathways. As you can see, it activates RAF, which goes on to be involved in the following signaling cascade. So MAPKs are a family of kinases that are well known to be involved in cardiac remodeling. They're involved in many vital cell functions, such as gene regulation, proliferation, and differentiation. There are 4 classical MAPK signaling cascades: the ERK1 and 2 pathway, as well as the ERK5 pathway, the JNK123 pathway, and the P38 pathway. These are all very complicated molecules with long, complicated names, but all you need to understand is that they're subtypes of MAP kinases that go on to phosphorylate downstream substrates. As we can see, there's a sequence of kinase phosphorylation that results in the ERK, GNK, and P38 pathways being activated. These go on to activate a number of downstream substrates, but most notably transcription factors, which go on to upregulate hypertrophic genes and increase protein synthesis and stability. If you can recall from the previous slide, this molecule, RAF, is activated by RAS, an upstream regulator. As the diagram shows, this activates the ERK2 and 1 pathways. RAS can also activate the GNK, P38, PI3K, and other intracellular pathways. And it has been shown that overexpression of RAS promotes pathological cardiac remodeling. As such, it is well evidenced that MAPK signaling has a very important role in cardiac hypertrophy and consequential heart failure. Overexpression studies have identified that simultaneous activation of ERK, JNK, and P38 after pressure overload contributes to pathological hypertrophy. These studies suggest that ERK activation plays an important role in the growth of individual cardiomyocytes, whereas P38 and JNK activation do not promote cardiac hypertrophy, but instead promote cardiac dysfunction, with JNK activation reducing gap formation and P38 activation promoting cardiac fibrosis. Altogether, these reduce diastolic compliance. So protein phosphorylation is a critical mechanism by which almost every cell activity is regulated, such as proliferation, gene expression, and apoptosis. Phosphorylation regulation consists of kinases and phosphatases, which act as a binary switch that regulates protein activity in an on and off state. ATP is hydrolyzed into ADP and a phosphate group by kinase enzymatic activity, and this phosphate is added to the protein. This phosphorylation of specific amino acids induces a conformational change that alters the protein's activity. Out of all 20 amino acids, only serine, threonine, and tyrosine contain a functional group that can accommodate phosphorylation. This phosphorylation may be activating or deactivating depending on the substrate and the site of the phosphorylation. Many proteins have multiple sites for this to occur that are regulated by different kinases, allowing the integration of multiple pathways through a common substrate. I finally want to talk a little bit about how kinases, particularly MAPK, have been targeted therapeutically. As we've seen, MAPK signaling upregulates proliferation and cell growth in response to stimuli. So the question exists whether anti-kinase therapies can be used in the management of pathological cardiac hypertrophy and heart failure, following the success of these drugs in conditions such as cancers. Mutations of a protein in the MAPK pathway can cause uncontrolled cell proliferation and so leading to cancer. The drugs that target this MAPK pathway typically inhibit these mutated proteins to prevent the uncontrolled cell proliferation and tumor growth. You can see in the diagram that these inhibitory drugs in red act at different steps of the MAPK-ERK pathway. With this evidence, it seems possible that similar drugs could be developed to combat pathological cardiac hypertrophy. So that's all from me. If anyone has any questions, please do put them in the chat and myself and Delvac can answer them at the end of the talk. And thank you very much.
Okay, that's really great, Neve. Thank you very much. So that's really a clear talk that makes my job really easy now because you have done all the background explanations about the signaling pathway, kinase signaling pathway, MAPK, and so on. So first of all, I would like to thank for the invitation and the opportunity to talk in this student society. So it's really great. So let me share my screen. Okay. Can you see my screen?
Yeah, we can see that.
Okay. So what I'm going to do this evening, I would like to tell the story that happened in my laboratory in Manchester in the past maybe 10 or 12 years. So it's based, it's a follow-up from what Neve has told us about how to understand the kinase signaling in cardiac hypertrophy, and then how we can use this understanding to design or to find a new target for therapy. But before that, just would like to little bit introduce my laboratory in Manchester. So we're interested in understanding the molecular basis of cardiac remodeling. As you know, the heart will change the structure and function in response to pathological stimuli, like what Neve has told us before. And there are a lot of processes that involve in this pathological changes is hypertrophy, the enlargement of the heart, fibrosis, development of scar tissue, apoptosis or cell death, inflammation, and autophagy. So we, in the lab, we interested in understanding how this process come together in the remodeling, and how we can find a new molecules that can be targeted to at least stop the process or maybe reverse the pathological remodeling. But this evening, I would like to focus on hypertrophy. I'm not going to talk about fibrosis, apoptosis, and so on, although this all also studied in my lab in the past 10 or 12 years. So why hypertrophy? Because you can see in this graph. So this is a paper published many years ago in 2001. This is a prospective cohort study in the US, I think. So they followed a cohort of patients with cardiac hypertrophy, and then there are about half of them treated with a drug that available at that time to treat the hypertrophy, and the rest were not treated. So there are group of patients that have a development of persistence of hypertrophy, and a group of patients that have a prevention of hypertrophy. And as you can see here, within maybe 3 years or maybe more, 5-6 years, you can see that there is a significant reduction of the risk of the development of heart failure and cardiovascular event during that time between people that have prevented of further hypertrophy compared to people that develop or persistent hypertrophy. So it is important that treatment of hypertrophy is important to reduce the risk of heart failure and improve the long-term prognosis of the disease. And then as you can see here, although at that time they can reduce significantly the risk, but it's not going down close to the baseline. Why? I tell you later. So this is just to follow up from what Neve has already told us before, that there are two types of hypertrophy: the physiologic hypertrophy. We're not going to talk about this today, but then the pathologic hypertrophy is the one that we interested. So pathologic hypertrophy is not normally reversible, it's irreversible. So it's induced mainly by pathological stimuli, and this is the most frequent causes of hypertrophy: chronic hypertension or high blood pressure, heart attack or myocardial infarction, and then subsequent neurohormonal activation. So this is a complex process that will activate, as shown before by Neve, a lot of molecules and signaling pathway that involve in this process. So then the problem is this: the current available treatment so far to control hypertrophy, if you have a patient with hypertrophy, is indirectly target this molecule. There is no therapy that directly target intrinsic cardiac factors. So the available therapy so far is targeting the extra cardiac factor. For example, reducing the blood pressure, controlling the hypertension, for example, ACE inhibitor, or controlling the abnormal neurohormonal activation like beta blocker and so on. So that's probably the reason why, when you remember the previous graph, it's not going to come dramatically to the baseline, the reduction of the risk factor, because we need something more powerful. we need something to target the intrinsic factor that probably govern or play important factor, play very significant role in the regulation of hypertrophy. So that's why we interested in looking at new target within this pathway. So I put a shadow here. This is the MAP kinase pathway that we are going to focus this evening. Okay, so now let's start with what we are doing in the lab. So this is a story that started maybe 12 years ago when I start my own laboratory in Manchester. So we have the idea that during the process of hypertrophy, the change of the heart from normal structure to hypertrophy. So at that time, there's already known that there's a lot of candidate regulators of hypertrophy. There are some factors, molecular factors and genes that considered as positive regulators or promote hypertrophy, and the other way around, there are some negative regulators, and both factors expressed in the heart. And then the theory at that time is that if one of these is overexpressed or the other way around, the negative regulators is downregulated, then there is an imbalance, and that will induce the growth and enlargement of the heart. So that's the theory that's already believed at that time, about 2005, 2006. But then we come out with the new analogy at that time. So we know that in other area in the medicine, so in the oncology, so we understand that probably there is something similar happening. So I believe that everybody knows the tumor suppressors and the role of proto-oncogenes in the development of cancer and tumor. So everybody knows that there are tumor suppressors in most of the organ or tissues, but it's also proto-oncogene. If there is any imbalance of the expression of these two, then there's a chance to the development of cancer. So then we think that tumor suppressor and proto-oncogene is also expressed in the heart. So that's what we found at that time, but we didn't know the function of these molecules, these proteins. So we have a hypothesis, maybe they do the same. Maybe the proto-oncogenes doing the same as a positive regulators of hypertrophy in the heart, and the tumor suppressor is the other way around. So this is the start of the program of work in my laboratory, that we start to look at tumor suppressors and proto-oncogenes in the heart, because there is no cancers known, or it's very rare. So we think that, or we hypothesize that this tumor suppressors and proto-oncogenes may do something different. And for example, it's probably play a role in the hypertrophic growth. So we interested in looking at this area because we know that there is a really interesting phenomenon in a disease called rasopathies. So rasopathies is a developmental syndrome that caused by mutation in genes along the MAP kinase pathway, the one that already explained by Neve before. So for example, this is the pathway. This is very simplified pathway. So if there is any mutation in any of these genes, for example, in one of the component in RAS, it's called Costello syndrome, in SHB2, Noonan syndrome, or the CFC syndrome, cardiocraniofacial syndrome. So the typical phenotype of people with this mutation is development of tumor or cancer, neoplasia, and then at the same time, development of cardiac hypertrophy. So there is a close link between cancer and cardiac phenotype. So then we think that probably we try to find something inside this pathway that probably can be identified or characterized in the heart. Okay, so we find one molecule called RAS-SF1, which interact with RAS and RAF and the MAP kinase pathway. Why we interested in RAS-SF1? Because this is a tumor suppressor. It's known to have a role in some cancer like bowel cancer and lung cancer. But when we look in the human heart failure patients, the RAS-SF1 expression is significantly downregulated, as you can see here. This is normal heart. This is from heart failure patients. And then when we try to induce hypertrophy and heart failure in mouse by doing TAC or transverse aortic constriction. So this is a surgical technique to constrict the aorta. So then the blood pressure is increased and then the mouse develop hypertrophy. So we can see a gradual decrease of RAS-SF1 expression during this model. 5 weeks reduce a little bit, and then after 12 weeks significantly reduce at the same level as in heart failure. So we think that it's probably play a role in the hypertrophy and development of heart failure. So then we try to find out the function further by performing genetic knockout analysis. So I would like to explain briefly. So in this approach, we try to mutate this gene in mouse. So we grow embryonic stem cells from mouse embryo and then creating a genetic construct that can delete specifically this gene, RAS-SF1, in the embryonic stem cells. And by some selection methods, we can find or isolate cells with knockout of RAS-SF1, the embryonic stem cells. And after we purify and characterize that it's really knocked out, mutated the genes, we put it back to the healthy embryo. And then this will create the, an offspring or mouse with a knockout of this gene. So in other words, this mice doesn't have RAS-SF1 in the whole body. So we have this mice, RAS-SF1 knockout, and compared with the normal litter mates, there is no difference in the heart size. But when we induce hypertrophy by generating or doing transverse aortic constriction, this one, putting a tie in the aorta, you can see that there is in the wild type in normal mice, you have a hypertrophy about maybe, I think it's around 30% bigger, but in the knockout, it's twice as big as in wild type. So it's the hypertrophic response is twice as much compared to the normal mice. When we look at the histological section, you can see here, hopefully you can see clearly, the cardiomyocyte is this one. This is the cross section, is bigger, is massive. So this is one of the biggest hypertrophic model at that time. So we published it in Circulation about 12 years ago. So then we try to find out, is it really affecting the MAP kinase pathway? So as you know, the hypertrophic stimuli, this is the MAP kinase pathway. So if activated by the stimulus, RAS, the upstream regulator of the MAP kinase, will bind to GTP, and this will activate the downstream signal. It will attract molecules called RAF1, which if activated will stimulate the kinase cascade, MKK1, ERK, and so on, and that's finally lead to the increased hypertrophic response or transcription or gene transcription. Then we to prove that RAS-SF working through this pathway, we generate a recombinant protein called RBD agarose. We generate a protein with a RAS binding domain and then link it to the agarose and the glutathione. So with the hope that if the RAS GTP or RAS is activated, this RAS binding domain will bind to the activated RAS, and we can easily detect by precipitating this protein, because there is a blue thing here. This can easily be precipitated and detected using Western blot. So this is the example, myoblastic cells. Just focus on the left-hand side. This is expressing an active RAS, or called RAS V12. This is a mutant active. And then you can see a really big lots of protein, showing that a lot of the activated RAS bind to this recombinant binding RBD agarose. And then at the bottom panel, this is not the this is cell lines, but this is a primary cells that isolated from rats. So this is more natural, and we induce these cells with phenylephrine, the adrenergic agonist. Then you can see that a lot of RAS is activated. But when we put RAS SF1A, you can see the difference. This is massive reduction of active RAS and significant reduction in the primary cardiomyocytes, meaning that RAS SF1A is really working by blocking this MAP kinase pathway. So what next? So at that time, we try to develop something. Can we target this molecule or this pathway for treatment? The answer is very difficult, because RAS SF1 is not a kinase. This one is not an enzyme. So it's really difficult to target this molecule. So we try to find other molecules that interact or in the proximity of this complex. So we find a signaling pathway called the hypo signaling pathway, in which one of the molecule called the MST2 is known to interact with RAS SF1. Hypo pathway is normally not related to the, not known at that time to be related to the MAP kinase pathway. But we, through the interaction with RAS SF1, we hypothesized at that time that it may also involve in the signaling through RAS SF1 and RAF1. So we generated the second knockout mice. We delete MST2 in mice using the technique as I shown before. And we did the same experiment, transverse aortic constriction. And the result is completely the opposite of the RAS SF1 knockout. So we see reduction of hypertrophic response from 50% to 30%. So it's 20% reduction. And also this is a marker of hypertrophy called PNP-ANP. So we are quite excited. We find a new kinase that linked to the pathway that we interested, and this may be targeted for therapeutic purpose because this is a kinase. So this published in 2012, and this is another mechanistic explanation. So first, we really want to know whether it's really MST2 linked to the RAF1 MKK or MAP kinase pathway. So we do the precipitation, and you can see it's really big, meaning that it's really tightly interacting. And then this, apparently, it's induced activation of RAF1. So MST2 is positive regulator of RAF1. So this is the evidence. When we overexpress MST2, RAF1 is and ERK1 downstream of RAF1 is phosphorylated. This is a Western blot showing the increased phosphorylation of this pathway. And of course, we knew before that RAS SF1 is included in this complex as a inhibitor of this pathway. But we are really interested with MST2 because this is a kinase. So we try to, at the time, we hypothesize if we have an inhibitor or a pharmacological compound that can disrupt MST2 function, we may be able to find new target for therapy. So this is just to summarize the findings so far, that we have RAS-SF1 as a negative regulator of this pathway. So then overexpressing of RAS-SF1 will reduce hypertrophy. But MST2 apparently is a positive regulator of hypertrophy. Then this can be inhibited if we want to reduce hypertrophic pathway. So we really need to find the inhibitor. And after a few years, we are really, our wait is over, because we try to find the inhibitor. We try to identify or design ourselves, but we are lucky because in 2016, a group in China, who is not the cardiology group, they work in liver, they develop, because they are really good in drug development, they develop a molecule that can inhibit MST2 activity. So this is how the design and the molecule work. So this is the MST2, and they design a compound that can disrupt the ATP binding sites of the MST2. So the protein won't work as a kinase. So this is the inhibitor called XMUMB1, and this is the inhibitor curve. And you can see that it's quite potent because the IC50, or the concentration to inhibit 50% of the activity, is in the nanomolar region. So it's 38 nanomolar for MST2. And of course, it's really difficult to have a really specific one. So it's also inhibit the other isoform of the enzyme, MST1, but at a higher concentration. So we thought at that time it's great. So we got this, we contacted them, and we got this XMUMB1, and we designed 2 experiments. First is in vitro, using cells, cardiac cells, cardiomyocytes isolated from rat, and then induce using phenylephrine to induce the hypertrophy, and then treat them with XMUMB1, the inhibitor. And then secondly, we also did the in vivo one. So we induce hypertrophy using TAC, constriction of the aorta. And then after a few weeks, we check the hypertrophy using echocardiography, but then we treated the mice with XMUMB1 for 2 weeks. And then at the end of this experiment, we evaluated the cardiac phenotype again. So this is the results. So it's just published 3 years ago. And this is the cellular data. As you can see, phenylephrine induced hypertrophy. You can see probably the cells bigger. Then after treatment with XMU from 1, 3, and 5 micromolar, we can see significant reduction in cell size. Of course, if you're interested, you can look at this paper. We did a lot more analysis to verify this finding. So a lot of molecular markers and other cellular studies that's been done to make sure that it's really anti-hypertrophic. Then this is the in vivo, the mouse. We did TAC, and then after 3 weeks, we did pre-treatment echo data to evaluate the heart phenotype. So using the echocardiography, we can measure the left ventricular mass using the imaging. So you can see that in the TAC group, it's really big, it's really hypertrophic, and wall thickness, the ventricular wall thickness is also significantly increased. Then we divided this mice into groups, half with treated with XMUMB1, half with vehicle. And after 12 days, we can see a trend of reduction of heart weight normalized to tibial length, but it's not very significant at the time. We probably it's the nature of the in vivo experiments. But when we look at the heart function, ejection fraction, you can see here that in the TAC group, the green and red is reduced before treatment compared to the sham or non hypertrophic mice. but the green one is the treated with XMU-MB1. You can see the improvement of ejection fraction after 12 days of treatment, and worsening of the function in the non-treated animals. And this also the same when we look at the end systolic diameter, also improvement in the green group here, the tag XMU, and worsening of the phenotype. Increase the diameter of the left ventricle means the dilatation. Although there was no difference in heart rate, but when we look at the cardiomyocytes histological size, we can see a significant reduction in the mice treated. And not only that, we also surprised that fibrosis is also reduced. This is a very important phenotype, the scarring of the heart. So it's really promising, but this is still in the preclinical stage in the small animals model. So I probably this is my last slide, so I can summarize my talk that from, well, it's only 20 maybe 10-15 minutes, but this is from 12 maybe more years of work. So we have identified several novel regulators of cardiac hypertrophy, which is linked to the kinase pathway, the major kinase pathway, the MAP kinase pathway. So we've got RASSF1A, the tumor suppressor, which is the anti-hypertrophic, and MST2, a protein kinase that the pro-hypertrophic. And then in the second part of the study, we have shown successfully that pharmacological targeting of MST2 can successfully control hypertrophy in vitro and in vivo in mice. And at the moment, we the project still ongoing. We collaborating with LifeArc. So this is a drug development company in the UK. They identified a new, more powerful inhibitor of the MST2. Because this one, the XMU, is not something that probably can be developed further because it's developed in other laboratory. But we have now our own inhibitor that we are going to test in hypertrophic model in mice. If this successful, of course, the next step will be experiments in large animal models like maybe sheep model or pig models before we can go to the clinical development in the future. So hopefully, we don't know. So I would like to stop here. This is the people in the lab involved in this project. So EFTA is doing all the experiments in MST2 inhibitor. Minzy and Sukpal help a lot in the developing the animal model and also doing the MST2 work. Arva is doing MST2 and a bit of RASSF1. And this is our collaborator that's helped a lot in making this happening. So thank you very much, and I would be happy to take the question.
I have a question actually. So the MST pathway, the inhibitor for it was actually developed for liver originally. And it got me thinking, how cardioselective is the MST inhibitor? Would it affect other organs or other areas of the body as well if it was used as a therapy?
Yes. So that's a good question. So of course, all of the MST2 in the whole body will be affected. Then of course, we have to look at the possible adverse effects in the other organs. For example, in liver, kidney, and others. So we did it. So it's published in the 2019 paper. Because we only induce cardiac phenotype, the liver and the kidney at the start of the therapy is a normal kidney and normal liver. And the result shows that there is no detrimental effects in those two organs after MST2 treatment. So of course, when the group in China also did the same experiments using liver failure model, and they found really improvement in the liver regeneration in their model. So my interpretation is that, of course, this will affect all MST2 in the other organs, but if it is not, if it is in the normal level, in the non-pathological level, it didn't do any harm. So that's what our interpretation. But of course, a lot of experiments need to be done to check the safety and also the off-target effects of this compound.
Thank you so much. That was really helpful. And another question from me as well, just since no one's asked on any yet. But in terms of your study, did you focus, just remind me, was it on just the different syndromes initially? So when you knocked the gene out, you focused on the RAS pathway that you had the knockouts in?
Yes.
Yeah. So would you feel like the indications would be different, or for all types, would it just be focused on genetic syndromes, or for all types of cardiac hypertrophy?
Oh, you're asking in relation with the rasopathies, the syndrome that I mentioned in the first. Yes. For example, the Noonan syndrome and also the Costello syndrome and so on, that's the people have got the mixture of neoplasia and cardiac phenotype. The reason I mentioned that is because that's the story, the beginning of the story. That phenomenon that happened in the lab, in the clinic, prompted us to hypothesize that there might be a close relation between genes that involve in cancer and in cardiac pathologies. So that's the reason why we interested in these genes, why we look at RAS-SF1 and also MST2 and so on, because these two genes, not many people at that time, 15 years ago, to look at the role in the heart, because this is known as a cancer genes. So that's the reason why we, I just show it as an example how we develop a hypothesis.
Oh, brilliant. Thank you for reminding me. I've understood it now. So in terms of your future research directions, where do you think you'll go from here?
Yeah, that's what I think I've already mentioned it. We try to find the more powerful inhibitor that we already have now from the collaboration with LifeArc. LifeArc is a company, this is a sister company from MRC. They did a lot of compound screening, so they got a library of maybe 100,000 compounds, and we just spoke with them, and they said that we've got some that probably fulfill our interest, inhibit MST2 10 times stronger than XMU-MB1. But they didn't disclose the molecule. They just gave us some of the compound, and then we now through a project funded by ReSearch UK, we now tested it in our mouse model of heart failure and hypertrophy. If it is successful, then the next step will be development in large animal models and probably clinical development. But that's the long way to go, because this will involve a lot more bigger collaboration.
Oh, honestly, it sounds fantastic. Thank you so much for giving such a brilliant talk. And if
You're
everyone,
welcome.
if you don't mind just filling out the feedback form as well, it'd be really useful for continuing on this sort of project for you all. And you'll get a portfolio certificate on completion of the form. And if you have any other questions either about Young Academics or specifically for Dr. Oceandy and Neve, please do let us know. And this recording will also be posted on YouTube later. Thank you so much for coming.
Thank you, everyone.
Okay, thank you.